Okay, have you ever stopped to think about how your body pulls off one of the most incredible engineering feats in the universe? We're going to unravel this amazing mystery of how it solves a completely impossible storage problem inside pretty much every single one of your cells. So, let's just start with a question. Imagine you've got something that's 6 feet long, about as tall as a person. Now, how on earth are you supposed to pack that? And here's the kicker. That 6 foot long thing has to fit inside a space so small you can't even see it. We're talking about the nucleus of a single human cell, which is way smaller than a speck of dust. It sounds absolutely impossible, right? But your body does this trillions of times over every single day.
That's right. The DNA from just one of your cells, if you could stretch it all out, would measure about 2 meters. That's a little over 6 feet. So how is this incredible feat of biological origami even possible? Let's find out.
So, this 6 foot problem, the challenge isn't just about cramming it all into a tiny space. Not at all. It's about keeping it perfectly organized, protected, and this is key, accessible. Without a system, it would just be a hopelessly tangled mess, useless. And that's where the real genius comes in. Think about it like this. A cell needs to use the information on this giant DNA map, but it can't create chaos by unfolding the entire thing every time. So the packaging has to be smart. It has to let the cell read specific genes when it needs to, and then pack them neatly away again. All right, let's look at the first level of this organization. We can call it DNA's everyday outfit. This is how your DNA is packaged during the normal day-to-day life of a cell. It's not the absolute most compact form, but it's the most functional one. This everyday state is called chromatin. And really, it's just a complex of DNA and some special proteins all wound together. You can think of it as the raw material of your genetic library, all nicely organized and ready for use. Now, not all chromatin is created equal. It actually exists in 2 states. You've got euchromatin, which is like an open book. Here, the DNA is loosely packed so genes can be read and used. And then you've got heterochromatin, the closed book, where the DNA is packed super tight to keep genes silent and stored safely away. And this ability to switch between being open and closed is absolutely fundamental. It's how a skin cell knows to be a skin cell and not a nerve cell, even though they both have the exact same DNA instruction manual. It's all about which chapters of the book are open for reading. So, what's actually doing all this wrapping and organizing? Well, it's time to zoom way in, down to the molecular level, and meet the microscopic machinery that makes this whole incredible packaging system work. Let's meet the heroes of our story, histones. These are special little proteins that act like tiny microscopic spools. Our long 6-foot thread of DNA winds itself neatly around these spools to get the whole compaction process started. And this whole process is just so elegant. First, 8 of these histone proteins cluster together to form a core. Simple enough. Then, a segment of our DNA strand wraps around that core almost 2 full times. This whole unit, the DNA and its histone spool, is called a nucleosome. And finally, another type of histone comes in, kind of like a staple, to pin the DNA to the core and pull all the individual nucleosomes together. What you get is a structure that looks just like beads on a string. Now, this beads on a string thing is just the first step. But wow, is it effective. It shortens the DNA's length by a factor of 7. And get this, a single cell has about 30 million of these beads. But we've still got a long way to go to solve our 6-foot problem. So let's zoom back out and look at the final, most condensed form DNA can take. This level of packaging is reserved for a really special and critical job, when a cell needs to divide and move its entire genetic library. And this is the chromosome. It is the ultimate travel suitcase for DNA. It's what you get when you take that chromatin string of beads and you coil it, then loop it and supercoil it again and again until you get these dense X-shaped structures that can be moved safely into new cells without getting tangled or broken. I mean, the level of compaction here is just staggering. From that raw 6-foot strand of DNA all the way down to this final chromosome structure, the DNA has been shortened by a factor of about 10,000. That is just incredible efficiency.
And if you look at a chromosome, you can see a few key parts. There's the centromere, that's the pinched-in waist where the duplicated copies are held together. Then you have the arms extending out, and crucially, at the very tips, you have telomeres. Think of them like the little plastic caps on your shoelaces. They protect the ends of the DNA from fraying.
And to bring this all right back home to us, this is the form our genetic information takes. In most of your cells, you have 46 of these carefully packaged chromosomes, organized into 23 pairs. They hold the complete blueprint for you. So, we've been on this pretty incredible journey, right? From a 6-foot-long molecule all the way down to a microscopic chromosome. But let's take a step back and connect all the dots. Why is this super intricate system of packaging so fundamentally important? Well, it really comes down to 3 critical jobs that are the absolute foundation of you. First, information management. It fits the entire encyclopedia of you into a microscopic library. Second, gene expression. By controlling which chapters are open, it tells a cell whether to be a muscle cell or a brain cell. And third, accurate inheritance. It makes sure that when one cell becomes 2, the next generation gets a perfect, complete copy of all the instructions. The stakes here could not be higher. When this elegant system of packaging goes wrong, if genes that should be silent are left open, or if chromosomes break during cell division, it can lead to absolutely devastating consequences, including cancer and other serious genetic diseases. This entire process, it's the foundation of your health, your identity, your very existence. And it runs with near-perfect precision in billions of your cells every single day. So the next time you think about how incredibly complex life is, just remember this elegant dance of DNA packaging, and maybe ask yourself, in a system this complex and this vital, what happens when just one single, tiny fold goes wrong?